Good morning. I am really excited to be closing out these TED-style talks and to tell you about the work that I did as a Virginia Sea Grant Graduate Research Fellow during my master's at Old Dominion University, and all of the really exciting lessons that I am continuing to learn from the temperate corals that live in our own backyard. But first, I want to take you here to a really beautiful tropical coral reef. And tropical coral reefs are pretty incredible ecosystems that support a wide biodiversity of life. And the biodiversity that is supported by this ecosystem is really built on a foundation of a symbiotic relationship. So if we zoom in on this one beautiful coral that we see here, we can tell that it's made up of many of these clonal coral animals. And corals are related to jellyfish and to anemones, so they can use the tentacles that you see here to capture food out of the water column. And that's one of the ways that they get the energy that they need to survive. And corals are translucent, so if we zoom in one more time, you can see that these orange brown smudges inside the tissue are actually many symbiotic algae. And so these algae and the coral live in symbiosis with each other. I'm going to call these algae symbionts for the rest of my talk. And these algae are photosynthesizing, so they're taking energy from the sun, they're converting it into sugars, and they're sharing that with the coral animal. And that's the main way that corals are getting the energy that they need to survive in the tropics. And so this healthy relationship between the symbiont and the coral is really crucial for healthy tropical coral reefs. And I think we should all care about having healthy tropical coral reefs because they do many great things, not just for the ecosystem, but also for us. So they provide habitat for a lot of the fish that we like to eat. They protect our coastlines from dangerous storm surge. And they also boost the economies of coastal and island communities through tourism. And unfortunately, as you might have heard, the world's tropical coral reefs are currently threatened by rising sea surface temperatures that are associated with climate change. So here we're looking at a really beautiful, healthy coral reef, and we can tell that it's healthy because the corals are brown, meaning that the symbionts are still living healthy and happy inside of their tissues. But just 1 year later, this same site looks very different. This is a result of increasing temperatures that have caused the symbiotic relationship between the coral and the algae to break down. And this process is called coral bleaching because, as it might be apparent from this photo, once that symbiont leaves the coral tissue, you're seeing through the translucent tissue of the coral down to the white skeleton underneath. And so this breakdown is really common now, unfortunately, throughout the world, and we're seeing sometimes annual coral bleaching events worldwide. And so I think the thing that surprises folks the most about what I do and when I talk about what I do is that we also have corals that live on the east coast of the United States. So here we are in Richmond, Virginia, and where you're sitting right now is not very far at all from one of the sites that I studied for my master's just off the coast of Norfolk, Virginia. And the other site that I focused on was in Narragansett Bay, Rhode Island. And the coral that I'm studying here is very special, near and dear to my heart now. It's called the Northern Star coral. And one of the things that makes it so special is that it's not required to live in symbiosis with these symbionts like most tropical corals are. So what I mean by this is that in the same site throughout its range, which is all the way from Cape Cod into the Florida Keys, we can find individuals of this coral that have symbionts, like this brown coral that you see here. And we also find individuals that are equally happy and healthy that don't have symbionts, like this whiter coral that you see here. And this is a really powerful study system because it allows us to disentangle the effects of the host and of the symbiont in things like responding to temperature stress, which is really important in the context of understanding coral bleaching and the effects of climate change on tropical coral reefs. And the other really powerful aspect of this study system is that because this coral lives across such a wide latitudinal range, it inhabits a really wide environmental gradient as well. So what we're looking at here is average maximum temperatures over the last 40 years or so. And you can see that this coral is living in places that are warmer, like in Virginia, and places that are colder, like my site in Rhode Island. And this really brings me to the broad question that I tried to ask in my master's, and one that I'm still interested in as a PhD student at Boston University, and that is, how does the environment drive coral responses to extreme temperature? And so to answer this question, I did a lot of diving in the mid-Atlantic, which I know you might be thinking that this is not as pretty as the tropical coral reef that I showed you earlier, but I do find these places to be pretty unique and awesome places to go diving. This is the wreck of the JB Eskridge. It was the site that I used off the coast of Virginia. And what you're looking at here, these arrows are pointing at colonies of the northern star coral that are growing on the surface of this shipwreck. And so, believe it or not, this is actually really lovely visibility for this site, and for most of the diving that I did, most days it looked more like this. This is me celebrating collecting a temperature logger in January on the wreck of the same ship, and took me only 15 minutes, don't worry, freezing hands in January. But I really bring this up just to highlight that this diving and this work, getting these animals to study them, was really challenging at times, but also very fun and rewarding. So once we collect our corals from Virginia and from Rhode Island, we would bring them back to the lab and run them through our coral stress test. So instead of a stress test for humans, where you maybe run on a treadmill, you're hooked up to some wires, this is our coral treadmill. So we seal our corals inside these chambers, and we're measuring their metabolic rate across a wide range of temperatures. And the point here is to ask the question of whether coming from a warmer home or a colder home affects your metabolic response to extreme temperatures. And this gives us an idea of essentially the thermal range that these different populations can inhabit and thrive in. And what we found might be what you'd expect or what seems intuitive, but namely that our Virginia corals are performing best at the warmer temperatures that we measured compared to our Rhode Island corals. So in other words, they have a higher thermal optimum than the Rhode Island corals do. And in contrast, our Rhode Island population performed best at the coldest temperatures that we measured. And this was a really exciting result for us and gives us a clue that these populations are adapted to the local temperature environment that they're growing up in. And so the next question that I wanted to address in my master's was, do genetics play a role in this observed pattern? And so the short answer is yes, but we looked separately at how the populations of the coral animal and the symbiont are connected or not. And what we found for the coral animal is that our Virginia and Rhode Island populations are connected. And coral reproduction is an entirely other talk, but you should definitely come find me later if you want to hear more about it, because I find it fascinating. But what I mean by these populations being connected is that coral babies are able to, once they're released, travel the currents long distances before they settle into a new home. And so presumably this connectivity is coming from a baby from one population making it and settling into this other population that we're looking at. And in contrast to the coral animal, for the symbiont, we found that they're distinct populations. And this is likely a result of the fact that the symbionts can't migrate as far, and so can't make that same kind of long distance travel that the coral babies can. And so the big sum of my master's work is really showing that this coral is very resilient to extreme temperatures across its range. And I really agree with what's been said a few times now, that this work has brought up a lot more questions than has answered. And so I'm continuing to use the northern star coral to do some of the research for my dissertation, including asking questions like, how flexible is this coral? So over the course of its lifetime, can it adjust its physiology to deal with things like increasing temperatures? Is this flexibility heritable? So can they pass it on to their babies? And finally, do we see the same pattern in other species? Because I think that understanding temperate corals like the northern star coral is really valuable and interesting in its own right. Ultimately, I want to be able to take this research and address it back to other tropical coral reef ecosystems, to apply this research to understanding better how these tropical corals are responding to climate change. And with that, I'd be happy to take any questions.